Good. Thanks for joining us today.
Oh, thank you. It's a pleasure.
We are really looking forward to your seminar.
There's been a lot of good seminars in this series. It's exciting to have this opportunity.
Do you have the YouTube link open?
I do not.
Okay.
Should I?
Yeah, no. Okay, brilliant. So before you start sharing your screen, maybe introduce yourself with a few sentences and then start your seminar.
So I'm an associate professor at Scripps Research Institute. I'm on the Florida campus, and I've been here about 8 years. I previously was in the McKinnon lab and did structural work, and our lab has been mostly interested in lipids and structural biophysical studies on the lipid membrane. So.
Thanks, Scott. Thanks. So thanks very much. So you can start sharing your presentation, and we're looking forward.
All right, can you see my screen?
Looks good.
Okay, well, we have all gotten accustomed to doing things online. This is not my first time online, but hopefully I get it right. Again, I'm really excited to have this opportunity. As I mentioned, we've been studying the plasma membrane, and recently we published a paper on titled Studies on the Molecular Mechanism of General Anesthesia. And that's primarily what I'll talk about today. We are interested in everything from pain and forced transduction and all kinds of anesthetics, inhaled anesthetics, alcohol. And generally we're interested at the biophysical level of how these things work. And as you can see here in a nerve, there's a signal has to come into the nerve and then get propagated down through an axon. And how those signals are controlled and what controls the excitability is in large part what we're interested in. And anesthetics obviously block pain, and they were first used back in 1846. And this is a picture of, it's called the Ether Dome at Harvard, where that was first demonstrated publicly. And since that time, there's been a question of how it actually works. These anesthetics are very diverse compounds. They're generally hydrophobic, and they range in size from xenon and nitrogen and even CO2, which are, well, xenon is a single atom. And diethyl ether, again, was the first anesthetic used, but there's all kinds of different combinations, and they're very just diverse structurally. And this has been puzzling since the very beginning of the diversity of anesthetic compounds. And it's led to a couple of competing theories. One was the membrane theory. It was first introduced over 100 years ago. Mayer and Overton independently discovered that these anesthetics, they partition into oils with increasing hydrophobicity. So on the y-axis, you can see here the concentration at which they're effective. And then on the x-axis, the propensity of the molecule to partition into oil. And the most potent molecules are the ones that are the most hydrophobic. And so given their diversity, and this was proposed that they were intercalating into membranes, and that somehow this intercalation changed the fluidity or changed the rigidity, the thickness, or something changed about it. And for literally 100 years, people tried to associate this membrane effect or this perturbation on a membrane with an ion channel or with some protein. And they simply did not succeed at that. There was no link between the membrane and how then there would actually be a loss of consciousness. And so about 30 years ago, Nick Franks in the UK and others began to propose this direct binding theory where an anesthetic would accumulate in the membrane, but then its effect was to directly bind to a channel and then gate that channel, either block a sodium channel or activate a chloride or potassium channel. But these proposals didn't get around the fact that there was this chemical diversity. And so it left this lingering problem. And people since that time have looked at anesthetics on all kinds of animals. One of the other problems is just the diversity of animals. So dogs, cats, elephants, like hippopotamus, mice, you name it, almost every single animal on the planet is inhibited or is anesthetized with all these different anesthetics. And the channels in these animals are dramatically different. And more recently, it's been shown that even the Venus flytrap, we're all familiar with this plant, right? When you're a little kid and you found them in the forest, it's really fun. You go along and you brush, you just tap the inside of the plant, and the plant closes in due to a mechanical response. Well, this year in January, a very detailed study was published on this by another lab. And you can see here's the plant. And if you touch, you can measure an electrical current. So this is, for those that aren't anesthesia, or are not necessarily familiar with electrophysiology, this is a current. It's just like what's in your plug at home where there's actually current moving through that can be measured. And if they touch the plant twice, they get two different spikes in current, and then the plant closes, the Venus flytrap, it closes. But if they take the plant and they put it in a bag of ether, this diethyl ether that knocks humans out, then that current goes completely away. And it's not even a little bit, it's like it's gone. And the plant, when you touch it, it absolutely does not respond to mechanical force, right? So plants diverged from humans 1.5 billion years ago. Like, think about that. That is a long time ago. And these plants don't even have any of the same channels as humans do. So it's really hard. And this is why this has festered this problem for a long time. This is just one example where there's many scientists in other fields who are saying, now, wait a minute here. Something has to explain this anesthetic effect. And these random direct sites seemingly have a hard time to explain this. And this was why it was a problem from the very beginning. So we were interested in it. Of course, one of the oldest structures of all life on Earth is the plasma membrane, right? We know this. Membranes and lipids existed from the very beginning. And so we came back to this 100-year-old question. Could there be a way that anesthetics could target the membrane? And if so, how? And the premise of everything that I'll be talking about today is based on lipid heterogeneity. This idea that lipids are not uniformly mixed in a plasma membrane. And I know this audience, for the most part, is very familiar with lipid heterogeneity, but there may be people on here that aren't familiar. So I thought I would take a couple of slides to discuss this nanoscopic lipid structures, or the lipid heterogeneity. And this actually comes from a review that Erding co-authored in Nature Cell Biology in 2017. And it very eloquently goes through a lot of the things that are really important to lipids. And I just have a couple of pictures here that I think are pertinent to what I'll talk about today. And that is, one, these ordered lipid domains. They're comprised of saturated lipids. They're long chain saturated lipids. and cholesterol organizes them. There's also this, what is called a disordered region, where the lipids are not thought to be as tightly packed. They're a little more fluid. And there's also cholesterol in these other domains, but it doesn't appear to regulate the function of the disordered region quite the same as the ordered region. There's a really important principle. This was discovered by Kai Simmons many years ago. I know Ilya worked on this and many in Kai's lab, is this fact that if you have a palmitate attached to a protein, it likes to partition or to hang out with these saturated lipids. And others have, tried to discover the biophysical mechanisms that keep these proteins in the clustered into these rigid domains. Mostly what I'll talk about today, though, is this palmitylation. And then my lab more recently has tried to combine another property of the membrane, and that is the clustering of PIP2 domains. We weren't the first ones to discover this. A group working with Stefan Hell showed that cationically charged proteins can help cluster PIP2 domains. Another group showed that PIP3 clusters and clusters separately from PIP2. And these domains are very different. PIP2 is primarily polyunsaturated. It's heavily charged. It has two to three phosphates on this sugar ring. And it generally is in a thin part of the membrane. And it also attracts proteins based on pH domains, SH2 domains, and PDZ domains. So you have this situation, and this is published, this is coming from a review I wrote with Carol Robbins. If you think about that, you have now two partitioned areas. One binds to PIP2, or a protein can bind to PIP2, or it can bind to these GM1 domains. And so this turns out to have an effect that we call raft-associated protein activation, or RAPA. And what this is we first discovered this, or described this. I mean, bits and pieces of it have always been known, and lots of people have worked on parts of this, but I think one of the very elegant descriptions of this was in 2016, where we showed that PLD, it's a phospholipase D2, it's palmitoylated. And so of course you would expect it to be driven into GM1 domains. But it also has a pH domain, a PIP2 binding domain. And this raises the question, well, where is PIP2 going to be? Is it going to partition with PIP2 domains, or is it going to partition with GM1 domains? And it turns out that if you apply, so it initially goes to PL, and I'm not going to go through all the data on this because it's published, but it turns out that it initially resides in GM1 domains. But if you apply mechanical force, it disrupts those GM1 domains just enough that now the PIP2 dominates. And so the binding affinity of the PIP2 causes PLD to then associate more with PIP2 domains. But its substrate, PC, and I know this is complicated, but this is really important to today's talk, its substrate is in the vicinity of PIP2. And it is this co-localization that then allows PLD to then be active. And once activated, it changes this PC into PA, and that's the signal. So, okay, to just kind of review that a little more, the mechanism that PLD is activated is not a conformational change. And many people think about enzymes being activated by some conformational change. And that is not the case with PLD. It is only activated, to our knowledge, by this translocation mechanism that exposes it to its substrate, PC. And that will become very important to today's talk on the mechanism of anesthesia. These domains are very close together. So we calculated, based in cultured cells, that these are about 40 nanometers apart. So the only way you can see this protein move in the membrane is if you use something that above the diffraction limit of light. And so we're using direct stochastic optical reconstruction microscopy, DStorm. It's a high resolution technique. I'm not going to go into tons of detail about it, but basically you have a blinking fluorophore and you measure multiple times the location, and then you take an average of that location, and you can get much more precise locations in a biological membrane. And these techniques were developed over the last 10, 15 years and are important to this study. The drawback is we have to fluorescently label, and there's potential artifacts of clustering. And I'll describe that more when I talk about the fluorophores we use. So this is just an example of the difference. These are neurons that we labeled, and we labeled PLD and PIP2. And if you look on a confocal microscope, they look like they're overlaid. So yellow is proteins that are in the that are co-localized or associated with each other. But then if you do the same labeling in DStorm, it became very clear that they are not overlaid. They're clearly around average of 42 nanometers apart, and that they're separated. And so the first thing we just wanted to know is, how do these domains behave in a biological membrane? And we did live imaging. This is about 100 millisecond resolution. And there are clustering of these lipids in general areas, and other people have seen this. And importantly, we see that the lipids exchange between these nodes, and they kind of exchange along a pattern. They exchange kind of on the graduate student, Nick, called them highways. Like they don't just randomly diffuse through the membrane. And you can see over a second, like that the individual nodes are loose and things are moving between the domains. So in agreement with what everybody else has said, we think that these are dynamic clusters. So how does anesthetics affect them? And the first really simple question is to just look at fixed cells, adding an anesthetic, and seeing what it does. And so this is looking at the actual size of the GM1 domains. It doesn't really tell us a lot of information other than maybe the anesthetics are affecting them. And this experiment is done in N2A cells. They are a neuronal blastoma cell line. And we simply added chloroform at a concentration that is normally used to activate ion channels in the clinic, which is around 1 millimolar. And we were very surprised to see that these GM1 domains actually increased in size and were larger. And the same thing happened with isoflurane. So this is statistical comparison of that. They were less than 100 nanometers to start with, and then they were substantially larger after anesthesia treatment. And this is in dramatic contrast to, say, methyl beta cyclodextran, which is a molecule that pulls cholesterol out of the membrane and removes these lipid rafts. So initially we were a little surprised. We were expecting the opposite. We were expecting anesthetics to disrupt the structure of the membrane. But nonetheless, this is what we saw. And it was disruption, but it appeared to be that they were getting larger. And we were looking through the literature. You're never the first one certainly to think of anything, and rarely the first one to actually get the first data on anything. So we assumed that others had thought of this, and sure enough, we saw this paper from 1997 where Richard Lerner had been trying to, he had found what he thought was an endogenous anesthetic with this oleamide, and he had described this this process where the ordered lipid domains were disrupted by this lipid that he found in the brain of cats. Anyway, it turns out Richard Lerner is at Scripps, and this started a wonderful collaboration with him. But we started thinking about it. Okay, maybe what's happening is what he's describing here, where where it's just disrupting the function of it and loosening up the ability of molecules to be sequestered into the GM1 domains. That would be described here in that maybe it was disrupted even though they look larger, and that PLD would be coming out. And we could test this, right? We could see, is the function of the GM1 domain actually disrupted? And that would be much more important than the change in size. So the way we do this is then we have to dual label. So we label the lipid, usually we're labeling GM1 domain, and then we label the protein. And then we do what's called a cross-pair correlation, where we statistically see how close are these things together. And if they're paired in this analysis, we see this kind of sharp decrease and flattening out. And if they're really just spread around and if there's not a real tight clustering at short distances, then we expect to see something like this. And normally you'll see in the data that I described, usually we'll take the first 5 nanometers and just describe at 5 nanometers, is there a change? And that allows us to statistically compare, is there a real change in how close things are together? So we did this experiment with PLD. It's a very simple experiment. You label GM1 domains with cholera toxin. Then we labeled PLD2, and then we did a merge, and then we used statistics to calculate what is the pair correlation of those two things. And again, remember, we expect the most significant pair correlation is at these very short distances. So are they highly correlated at less than 20 nanometers? And so this is our control right here. And we see this is a unitless number. It's just correlation of association. And we can see with the control that PLD is highly associated with GM1 domains. If we add methyl beta cyclodextrin and we get rid of those GM1 domains, well, of course, PLD association goes down with GM1 domains. Interestingly, though, if we add chloroform or isoflurane, we also see this very dramatic decrease in association with GM1 domains. So how do we interpret this? This is just a statistical calculation of that at 5 nanometers. So, so we conclude from this is that PLD is associated, but then when you add an anesthetic, it's leaving this raft and presumably going to PIP2 domains where we had seen it go before. Remember, these rafts are actually getting larger in size and more of them, and yet PLD is still leaving. So that was significant to us. Well, what could PLD be doing then to control an ion or to induce anesthesia? And we work on this channel called TREC. And TREC is a 2-pore domain potassium channel. It's known to set resting membrane potential, and it's activated by inhaled anesthetics. It also is activated by mechanical force, and it attenuates pain, which you would expect for an anesthetic sensitive channel. Interestingly, it's activated by PLD. And the way it's activated is the product of PLD, this phosphatidic acid, binds, this is a 3D structural representation, or crystal structures of the atomic structure in active and inactive states. And this helix moves up and down, and this lipid influences this helix moving up and down. And I'm not going to go into detail exactly how it's activated, but more or less that's the situation for activating TREC. Interestingly, PLD, this enzyme that I just described that comes out of GM1 domains, binds to the C terminus of TREC. So this raises a very important question. Is the anesthetic really binding to the channel? Remember, everybody was assuming that the way anesthesia worked was that it was going to bind directly to a random site, just some random site in the transmembrane domain here somewhere. Remember, because anesthetics are really hydrophobic, they're sitting in the membrane. So they would bind to some random site here and then activate TREC. That is what had been proposed. People have been studying this for 15 years. But now we found that the enzyme that activates PLD is activated by anesthetics, is anesthetic sensitive. So this raises the question, which is it? Is it both? Because they're not mutually exclusive, right? Do anesthetics bind directly here? Or is PLD the anesthetic sensitive protein, and is the real target the membrane, right? And this would be important because nobody had ever shown a way that you could target the membrane with an anesthetic and then have it affect a protein. So, so this is the model we set up then. If anesthetics are disrupting, the PLD would then come out and activate TREC1. So can we test that? So this is a very important experiment, and so I'll go through it in detail. PLD, there's a known mutation in PLD where there, in the catalytic site, there's a single arginine mutation. And if you make that single point mutation, the enzyme doesn't catalyze any lipid synthesis. It's dead, catalytically dead PLD. Otherwise, it's identical. And we reasoned, if this anesthetic is binding to the transmembrane domain and activating it directly, and we put in a catalytically dead PLD, the channel should open with or without PLD, because it would be the direct binding that was opening. If PLD is the anesthetic sensitive channel and it's these lipid rafts that are the target, then it should be required to have active PLD for TREC1 to be open. So we did the experiment. We expressed TREC1, and this is the only experiment that's not done with endogenous TREC1, and that's because we needed a system where we only had TREC channels so that this current would be isolated to TREC. So this is done in HEK cells, overexpressing TREC, and then we add chloroform, and this had been seen before when you, so let me back up. On the y-axis here is current. So this is the amount of, this is just like the current that I showed on the plant. It's an electrical current, and we changed the voltage here. And as you increase voltage higher and higher, the current increases more and more. And this kind of gives us a sense of confidence that, yes, this is a real current that's voltage dependent. And when we add chloroform, you can see that the current significantly increases. And this is what had been shown 15 years ago by Patel. Everybody knew that this channel is anesthetic sensitive, and this is why, because when you add chloroform, the current goes up. So now we do our XPLD experiment. And remember, and we see that there's absolutely no current at all when you have a catalytically dead PLD. And so this demonstrates that the activity of the channel is completely dependent on PLD being there and on this lipid being produced. And it shows that even if the anesthetic can bind the channel, it's completely dependent on disruption of the lipid raft and PLD coming out, because you have to have PLD. It's an absolute, it appears to be required, at least in these conditions. So this strongly suggested that, hey, maybe this membrane really is the target of anesthesia for TREC's response. And this is just controls and showing statistically that these currents are real. But these are very robust signals. The channel is clearly anesthetic sensitive and disrupted by PLD mutant. So we would predict if this is a general mechanism, then PLD should be activated by all these different types of anesthetics, right? The membrane, if the membrane is the target, the membrane could react to all these different molecules. And if it did, then it should activate, they should all activate PLD. So we developed this live PLD assay. So PLD is just expressed in a cultured cell. And when PLD is activated, it releases choline. and then we convert that choline to a fluorescent signal. So in red is inhibitor, so when PLD is inhibited. We always see basal activity of PLD, which is in blue. And then when we add an anesthetic, we can see increased PLD activity. And so we saw very clear activation by both chloroform and isoflurane. We did this compound called F6, which is a volatile molecule that disobeys this Overton-Mayer. It doesn't seem to activate, at least not at the concentration that Overton and Mayer predicted. It doesn't cause anesthesia. And so we used that molecule as well, and we didn't see activation of PLD. And so this gave us a lot of confidence that the mechanism that, and this is just a summary of all of them. So we did diethyl ether, chloroform, isoflurane, propofol, again, this single atom, xenon, they all activate PLD. Ketamine is an anesthetic, but it's thought to be rather specific to glutamate receptors. So we didn't expect that. And then the F6. And so this demonstrated that all of these anesthetics are likely disrupting the membrane, and they all clearly activate PLD. We did a dose response curve using this PLD signal in live cells, and we see low millimolar KD for chloroform, which is the same that people see with the ion channels and is expected for clinical effect. And lastly, it was not known that propofol was an agonist of TREC. And so we said, hey, could this assay predict the molecules that will activate TREC? And sure enough, when we put propofol in an electrophysiology experiment with TREC, it activated the channel. So we concluded from this that the membrane is a bona fide target of anesthetics. We also wanted to test, kind of as a control, are the anesthetics directly binding to the channel? And could it, you know, if we purified the channel, could we see some type of activation? We were not able to see activation. This is so we purified TREC1, we reconstituted it into lipids of known composition to activate it. PG is a lipid activator, so we added a half maximal amount of PG, so we get a little bit of activation in this control, which is this TREC control right here in gray. And when we add propofol and we add chloroform, we see absolutely no activation, any additional activation. So the channel is halfway activated, and we cannot see additional activation. And we used a cis loop gain of function where we make a disulfide here and we trap it in the up state to get full activation. And we can show that the system is capable of more activation, and we just don't see it. So it's a negative result in the fact that we don't see activation, but this is a really important experiment. For a long time now, ion channel people, which I am one of them, had been claiming that anesthetics could directly activate a channel, but we hadn't done these experiments where we purified the channel and put it into a purified vesicle and could directly test it. These experiments need to be done to demonstrate that there's direct interactions. And so this supports the idea that no, the anesthetics don't appear to be binding directly to the channel. Again, somebody could come and show that they are. I'm not saying that it's impossible that they couldn't, but so far we have no evidence that there's direct binding of these anesthetics to this channel. And so PLD is likely the mechanism. We also did local anesthetics. Local anesthetics are known to inhibit TREC, and we did see inhibition of TREC. So this with local anesthetics. So the inhaled anesthetics. We can. This is a system to see activation and inhibition, and the fact that inhaled anesthetics don't do anything is highly supportive of the model for PLD activation. Okay, so there's one other experiment that is critical, And that is to convert TRAC to an anesthetic sensitive channel. So TRAC is a homolog of TRAC. And it's been known for a long time. We repeated it. These are experiments where we repeated it. It's known that when you add chloroform to TRAC, the channel doesn't do anything. This is at plus 40 millivolts, and it just sits there. It doesn't, you add chloroform and it just doesn't do anything, right? But we knew that TRAC is a homolog, and we knew that it was activated by phosphatidic acid. So we devised this experiment where if we take the PLD binding domain from TRAC and we swap it for TRAC and leave the transmembrane domain the same. So this is TRAC, transmembrane domain, and it just has this little piece of TRAC in the C terminus. Would this convert TRAC into an anesthetic sensitive channel? And surprisingly, it robustly does. So this is TRAC with this little piece of the C terminus. And now when we add chloroform, 1 millimolar, the same as TRAC, we see almost more robust activation than we saw with TRAC. And of course, it's PLD sensitive. So if you take out the PLD, it no longer activates. So, and this is just statistically, and this isn't, you know, I mean, it's very, very robust. So this isn't, you know, squeaking out a little bit of anesthetic sensitivity. This channel is clearly anesthetic sensitive. And it suggests that, that this motif is modular and could be moved around, and anesthetic sensitivity moved to other molecules. And so this supports this model where the lipid raft is the target of anesthesia. And I really think that this is the key experiment, that this is very difficult to explain without a membrane-mediated mechanism. If the membrane isn't the target of anesthetics, how then is TRAC responding just with this little disordered loop on the C terminus? I really think this is the experiment that really nailed down that the membrane is a target of anesthesia, combined with the imaging of the lipid rafts. Of course, we wanted to show this in an in vivo model. This is, so a student in my lab devised this experiment where he 3D printed this plastic chamber, and then he pushed the gas across the chamber. And then he used a camera with the help of the Jaw Lab here at Scripps to image flies moving around. And then as you put anesthetic on the flies, they drop dead. They don't drop dead, but they look like they drop to the bottom and are completely out, right? And we can then measure that. And we made a PLD knockout, or we didn't make it, we obtained a PLD null fly. And then in theory, if it's important for anesthesia in the whole animal, it should be resistant to anesthesia. And that's exactly what we see. So at this concentration, the flies pass out. These are done side by side in the same chamber. So we're doing, we're applying the exact same anesthetic at the exact same time. And we can see almost all the wild type flies are passed out before any of the PLD flies pass out, and showing that the fly is resistant. And those are dose responses. Anesthetics have very steep dose responses, but we statistically can see that there's a difference in that response. So we wanted to image these domains in the actual brain of an anesthetized fly. And this is just, so we use super res imaging. We anesthetized the fly, or we took a fly brain and then put it in chloroform, and then did this exact same control experiment that we did in tissue culture. And we could see that the domains in the fly brain increase similar to what we saw in culture, albeit not as dramatically. And so we concluded from this that this 100-year-old question, and I think the paper generated considerable amount of attention. And I think it's partly because this is a 100-year-old question. is the membrane a target? And I think we have to say yes. Now we know that the membrane is a target of anesthetics. And this will have implications, for example, on teaching medical students. For a long time now, they've been saying that the membrane wasn't a target. And even medical board exams generally have a question alluding to the fact that the membrane is not a target. And those exams are going to need to change. And how this is taught in schools is likely going to need to change. I don't think it's going to change how an anesthesiologist right now is going to practice medicine, but I think eventually it could have a change based on the things we learned from this. How far-reaching will this be? There's lots of, all this system requires is palmitylation, which we know there's lots of proteins that are palmitylated, and then a PIP2 binding domain. And if we just look down through the number of channels that have this criteria, for example, GABA A is a very important anesthetic channel. It is known to bind PIP2, and it is also palmitylated on the gamma subunit. And it has the exact same properties that PLD has. So we would expect that a channel could also move in and out of these domains. And then this is just a list of just a few of the channels that have these same properties and where they're located in the different subunits. So obviously this needs to be investigated on multiple channels. And how would this work? This is just a model of how you could get anesthesia based on this lipid membrane principle, right? On this function of the membrane. And if one lipid, if a lipid activated, if the same lipid activated a channel and then inhibited another channel, then moving the channels in and out of these lipids could have the opposite effect on, and it's probably a little complicated to explain, but it's known, it's known that all the GABA A channels are activated by all the anesthetics, all the glycine channels, and these conduct chloride. All the chloride channels are activated. Almost all the sodium channels are inhibited, and they're inhibited by all the different anesthetics. And this is very puzzling. Like how can all the sodium channels be inhibited and all the chloride channels be activated, and in all the animals, all the way to plants, right? Like it just it seems really bizarre that you could get that combination. And so this model here, and I'm not going to go into detail over it in time, but using a lipid intermediary and using the membrane as a mechanism, we could explain how all the calcium channels and sodium channels could be inhibited and all the chloride channels activated. And Erdinch, I had 3 slides on COVID-19, but we've gone pretty far. Should I just end right here?
This is up to you. We have a lot of questions already. If you want to show your slides, go ahead and show your slides. But if you want to answer the questions, we can also stop here. Up to you.
Yeah, just have, so yeah, I think I'll show them then. It'll take less than 5 minutes. So remember now, the plant diverged from humans 1.5 billion years ago, right? And this suggests that there has to be something very fundamental to the cells. And so we're arguing that it's the membrane. But if there's something that's that fundamental that it wasn't obvious enough to understand anesthesia, the membrane-mediated mechanism of anesthesia, this mechanism is probably working in lots of other things. This is a fundamental thing to the membrane. And I think this is an example. I think these lipid rafts and what Kai Simmons was proposing and these other things that have been coming out over the last 15 years, I think that we're going to see that these are very fundamental things to the membrane, and that there are very important questions that physiological questions that are confusing right now because people haven't paid attention to this science that's been going on, and which is why I love this this series, because it's had a lot of those people that actually are paying attention to this. And I think that this is going to show how important it is. One of the things we did when COVID-19 came out is we realized that this ACE2 receptor is sensitive to cholesterol, and that it is probably moving in and out of rafts too. And one of the things that was known about COVID-19 was that obesity is very bad underlying condition. It's known that if you get older, that you're much more susceptible to COVID-19. And so we showed that the virus entry was dependent on this cholesterol. And then we imaged ACE2 going in and out of the lipid rafts. And we could see that when you had high cholesterol, that the SARS-CoV-2 virus was clustering, and that in young people and people who were otherwise healthy, the ACE2 receptor was outside of the rafts, and so they would be protected from infection. And we did this in a mouse too, where we fed mice a really bad diet, which is what most Americans eat. And we see the cholesterol go up in the lung of those mice. And then when we looked using super res imaging, we could see that the ACE2 receptor was positioned for endocytosis and bringing the in these diet-induced obesity mice. So it was very pronounced in an old mouse that had diabetes. And this was a model that we proposed for how the virus could be very sensitive for old people that are diabetic or that have other underlying conditions. So I think obviously anesthetics didn't evolve to target, they didn't evolve so that a doctor could cause anesthesia, right? And clearly the plant didn't evolve to be sensitive to an anesthetic. There has to be endogenous mechanisms that are important to biology. And I think this is just one example of where these types of things are going to be broadly involved. So with that, I will finish. Again, we conclude that the membrane is a target of anesthesia. I want to make it clear that we do not think that is mutually exclusive with direct binding. This was a huge mistake of people who were studying direct binding, that they said because there is direct binding, there can't be a membrane effect. And there's no data to really back that up, that you couldn't have an anesthetic effect. And then I think that these cells are harnessing this lipid heterogeneity to gate channels. And I think in the future that we will find endogenous analogs of anesthetics, and we'll need to address some concerns about stereoselectivity in ordered lipid domains. And this is my group right now. Arif did most of the stuff in the with the anesthetics in the PNAS paper. Nick developed a lot of the techniques for the PLD assay. And Zishwin did COVID-19, and Zishwin is working on anesthesia as well. And I can't thank Richard Lerner enough. He's a fantastic collaborator. Since the day we started working together, he's been a continual support for this project. Keith Miller, also here, was here at Scripps, is now at Stanford. He helped develop the imaging for the flies. And we were funded by a DP2 award from the NIH, and now an R01 in funding from the Department of Defense. So with that, I will stop and happy to take questions.
Thanks a lot, Scott. Wonderful. So we have a lot of questions, but by tradition, I start with my own question, just because I can.
Should I go on the slides, or do I stay on the slides?
No, you can stay. Maybe you can refer to some slides later.
Okay.
So I'm pretty sure that you're aware of Sarah Veach's work of anesthetics and critical temperature of the membrane phase separation. How would you reconcile these two things, PLD and the critical temperatures?
So yeah, Sarah Veach has done wonderful work on especially these GUVs, the budding from off the cells. And we do think temperature is really important. I actually, my response to this is actually a conversation I had with Richard Lerner, because I asked Richard Lerner the same thing. And he thought that because temperature changes very quickly, right? And the question is, how is this not being affected by temperature? And his response is that biology adapts to the problem it has at hand. And so from the very beginning, there's always been changes in temperature, and sometimes very quickly. And so biology would need to adapt to that. And how does it? He suggested it does it with desaturases. So, and if you look, if you look in the cell membrane, desaturases work very quickly, and the body and organisms do have to adjust to temperature. And I think what Sarah is seeing is very indicative of a real effect. The problem is when the enzymes are in control and they react very quickly, you have to block those enzymes and you have to study it with that disabled in the cell. And I don't think that's been done yet. We would like to do that. That is something that needs to be done. And I think once you inhibit the enzymes that are changing the fluidity to compensate for temperature, I think you will see an anesthetic effect. And I think you can already experience it. If you get your hand really cold so that the enzymes can't adjust, and then you warm your hand up really fast, you do experience pain, like the nerves fire. And I think that is kind of what Sarah Veach would indicate, based on her system. She has great experiments. We have had a hard time. When you overexpress a protein, so two things. Sarah is budding off these GUVs, and PLD, any perturbation to the plasma membrane and PLD leaves. So we haven't been able to bud it off. We can't, it's hard to form them without really, that disrupts the membrane quite a bit to break those GUVs off. So we haven't done those experiments. We've thought about them. We would love to mix that. And the other thing is we can't overexpress proteins. This RAPA, this RAFT-associated activation mechanism, when you overexpress a protein, it goes everywhere, and it just completely, it goes into RAFs, it goes into the disordered region, it's on PIP2, it's just everywhere, and it destroys the native regulation. So that's how we ended up with the set of experiments we have.
Okay, so one more short question. So when you have the anesthetics and you see that the PLD is leaving the GM1 domains, are you also sure that it's actually going to PIP2 domains?
Yeah, well, we've done it now with mechanical force, and yes, we co-label the PIP2 and we show it go to the PIP2 domain, so it increases on the PIP2 domain.
Okay.
Yeah, it's just the person who was doing PIP2 domains wasn't Arif, the one that I've said many times. I know we believe this is happening with anesthetics too, but we need to confirm it in anesthetics. We haven't actually done that. The ACE2 receptor, so everything else since then, it's kind of a rule in the lab. Yes, you need to look at PIP2 as well. Okay. So ACE2 is going to PIP2 domain when you decrease cholesterol. And some of the other ones we're studying as well that we haven't published yet.
Sounds good. So I will start with the questions by the audience. I must say you're quite popular in Turkey because there are a lot of questions in Turkey. I think you probably have a visitor at some point in your lab. So the first question is by Enes Akyüz from Turkey. He says, considering the effect of anesthetic agents on GM1, an increase in raft size was clear. If we think of lipids as a bicycle shock absorber, can we say that this increase can also affect the KIR channels in PIP2?
So well, there's lots of KIR channels, and I do think some of them will be palmitoylated and maybe interact with GM1. And the GM1 domains are the ones that are most affected, to our knowledge, by anesthetic. So if the KIR channel is associated with the GM1 domain, I would say yes. And we've started to look at that, and we have some evidence that there is some association with a few of them. The question is, though, with PIP2 domains, because that's what really KIR depends on for its activity is PIP2. And I don't think it will be dramatic just with PIP2. PIP2 is affected a little bit because I think we'll find out more as we study it more, but I don't think anesthetics are going to be as dramatic on proteins that only bind to PIP2. It needs both. You have to be going into GM1, and that's what we mean by this RAPA mechanism, that it's associated with GM1 and it leaves to get activated.
Okay, next question is by Arda from Turkey. Do you have any other lipid or molecular candidate that has a role in the anesthetic agent's effect other than GM1 and PIP2?
So PIP3 is, so PIP2 is a arachidonylated, meaning it is highly unsaturated. PIP3 is short chain saturated, and it's clearly separate. So we know of at least 3, right? I think that there's going to be many domains. I think we don't have evidence other than PIP3 to answer the question, but I think that there's going to be other domains. And I think that combinations of sugars, so sugar binding proteins with different lipid acyl chains, I think you're going to get these aggregates of places to sequester proteins to turn them off. And who knows who will find those and how we'll find them. There's a wide open arena there.
I mean, related to this, there are also some findings by Martin Hoff, who believes that actually GM1 domains, they form domains regardless of the cell. Even if you reconstitute just a few mole % of GM1 in a GUV, they will still form domains. But these domains may not be ordered domains. They may not be rough domains. But the ones that you think in the cells that you're basically having PLD2 associated, they're definitely ordered domains, right?
Yes, and I think the distinction between the ordered domain and the other types of domains, like these sugar domains, like PIP2 is a sugar, right? Is that palmitate, I think, is dependent on the order. And we have more evidence that we have some other papers that are going to come out that will substantiate this with data. But I think Kai is really the one who put this forward. That is a very important interaction, and it's the palmitate with the ordered domain, right? And so I think that domain is very unique. It could be the only one. Maybe there's some others, but I think that one is much more unique in the fact that if you're a palmitoylated protein, your address, your target site, where you're headed, is probably going to be GM1 domains because of that order. Now, when I say that there's other domains, a different sugar could be in a different domain. And if there's a binding protein, like a pH domain that's specific for that other sugar, then I think it will pull it away from the GM1 domain and to that other location. And there could be another enzyme in that other location, and you could get a completely different activation. But I do think that kind of the central or the main player or the kind of the glue that holds it all together, the thing that's really, really important is the GM1 domain. And I think it's unique in that property.
And with the anesthetics, PLD still has the palmitylation. Is that correct?
Yes. Yeah.
Okay. So anesthetics wouldn't change the palmitylation.
So we would conclude, although we haven't shown with absolute certainty, we would conclude that the anesthetic was disrupting the ordered interaction with the palmitate and the GM1 domain. That whatever that ability is to get that palmitate to have affinity or to attraction to the GM domain, that is what's being disrupted by the anesthetic.
Okay, next question by Christina. And she's asking, can we think of difference in blocking in Treg1 as the outward positioning of the lipid that controls ion channel activation?
Whoa, say that again.
Okay, can we think of the difference in the blocking in Treg1 as the outward positioning of the lipid that controls the ion channel activation?
Which ion channel is it? What's the ion channel?
The TREC1.
Oh, TREC1. So, okay, sorry, I didn't know what channel it was. So say it again, I'll get it this time. I didn't know what channel you were talking about.
Okay.
Can you repeat it one more time?
Okay, so I'm repeating again. can we think of the difference in the blocking in track 1 as the outward positioning of the lipid that controls ion channel activation?
So I'm really biased. I like lipids a lot, right? Like, I'm a lipid guy. So you're asking the wrong person. That's a huge controversy. I think the lipid is what is controlling the channel. And I think that, yes, I think that when that lipid is there, the conformational change is induced by the lipid, and it's a lipid-gated channel. Mm-hmm. Other people might think differently, though.
And a follow-up question, do the effects of the anesthetic change in diseases with ion channel mutations?
So in disease?
A disease that would involve ion channel mutations.
Yeah, so that's one of the things. There are individuals who have different sensitivities to anesthetics, but I don't. And there are some cases where somebody might say a person is even resistant to anesthetics. Is it complete or not? So I do think that individual mutations on ion channel, the ion channels are in our model, the ion channels are the downstream effectors. They're not the primary targets of anesthetics. But that doesn't mean if you have a mutation in an ion channel, that's clearly going to affect excitability, and there's going to be a change even in anesthesia, I would predict. So yes, but let's take the GABA channel, for example. You can remove the GABA channel completely, and that will affect humans because they need the GABA channel. But if you go to, say, a plant that doesn't really have a GABA A channel of any sort that we know of, it's used to functioning without it, and it's using the lipid membrane. It's using these lipid rafts, this functional, this biological functional thing in the lipid membrane. It's using it for its own purposes. And it's always been there. And that is what's not going to go away. And so even if you have a mutation or you have a different species or something else, that's why we think the anesthesia is so well conserved.
Okay, next question by Sarah Keller. On your first track 1 data slide, why is there significant current when there is a mutant PLD in the presence of chloroform? Should I expect zero current instead?
No, because which slide? Let me go back up to that.
It's the first track slide.
First of all, no, there should not be no current, and there's multiple reasons why. One, so in this slide, no, there's always basal level of track in all of our experiments, and that's because one, there's always endogenous PLD. Even when we add the catalytically dead, there is some endogenous, and we are assuming that it's, there's a binding site on track for PLD. So if you overexpress PLD, then there's more catalytically dead PLD around, and it competes off the endogenous. The other thing is there's always a little bit of background track because PG, it also activates it. And there's other sources of PG other than PLD. So there's probably 2 or 3 lipid modifying enzymes that contribute into track, but the anesthetic sensitive one that is the most prominent is PLD. So I don't know, did that answer it?
Yeah, I think so. Thank you, Scott. The next question is by Betul, saying that the ion channel structure is in angstrom and your analysis is nanometers. Would it be sufficient? But I think they're 2 different things. Is that correct?
No, but we have thought about these distances, and they're very consistent. So the movement of that helix up and down is about 10 angstrom. So that could happen very rapidly. It's just a conformational change. The distance going from a GM1 domain to a PIP2 domain is only 40 nanometers. So that could happen in about 500 microseconds. And it's actually a very important point that it's only 40 nanometers apart because this is faster. The diffusion is faster than electrophysiologists can measure using electrophysiology. So it's been missed because it's such a short distance that that you can't see it in electrophysiology. It appears instantaneous by electrophysiology.
Thank you. AJ is asking, you never mentioned GM1 in your 2012 Nature article. Is this a new discovery? No, it's not. Do you think it is not enough to just think of PIP2 for Q channel activation?
I don't think it's just enough to think about KR2. I think that even if KR2 sits in PIP2 domains, there's probably a PIP2 modifying enzyme that could be controlled by GM1 domains. So, or for example, PI3 kinase or P10, any of these enzymes could be controlled with GM1 domains and RAFT-associated protein activation. So I think, again, this went back to why I was describing COVID, right? Like these mechanisms are probably widely applicable to all kinds of things.
So one question I have is, when you show the effect of different anesthetics, they have different extent, right? So the effect is not exactly the same.
Yeah.
Can you explain this chemically with the association to the membranes? Well, or another, just a follow-up of this.
Yeah, no, that's a very good observation. And it's actually even more interesting than just in that one cell type. As you move different cell types, the ratio of the anesthetic, the potency of the anesthetic changes too. So propofol is much more potent in a muscle cell than it is in a neuronal cell. And do I know what's causing that? I don't, but I could guess. And I think that the different types of lipids in the GM1 domains are probably not exactly the same. So what could change? The amount of cholesterol could change slightly. The actual linkage or something on the lipid itself could be slightly different. The chain length, so the long chain sphingomyelins in a neuronal cell might be slightly different length than in a muscle cell. And then how those anesthetics pack, it might pack a little bit better and a little bit, one might pack a little bit better in one and one in the other. And then when you switch the lengths, the longer length could accommodate one and not accommodate the other as well. And I think this ordered packing has very, one of the reasons I think it's so important is because I think the energy difference is very, very small. In other words, to get that palmitate into that domain, I think is a very, very small energetic barrier. And so just slight changes in that, I think, is what the cell is using and has fine-tuned. And I think that with all anesthetics in all different cell types, you're going to see very different reactions. It would be my prediction.
And one follow-up. So you showed this 100 years old graph by Overton that anesthetics, the liquid, the oil-water interface, they basically go to the hydrophobic parts. But there are this, as far as I know, there's some new anesthetics that deviate from this.
Yeah, so that was, yeah, that's this one right here, the F6.
Yeah.
It's not an anesthetic. So yeah, it doesn't lie on that curve. You know, it's remarkable that there's only a handful of molecules that anybody's ever found that don't lie on that curve, right? So my response would be, it's actually marvelous that there's only 4 or 5 of them and they're all kind of similar and they have some weird aspect to them that we don't understand. We're doing a follow-up on this. I don't know that I'm ready to explain. We understand F6 now a lot better, and we'll follow that up. But yeah, they are outliers, and and the fact that there are outliers in this system too, I think is a very strong indication that this system is involved in anesthesia.
Okay, next question is by Hussein Amre, saying that while hypoxia is observed in some epilepsy model experiments, some are not. Could this contradictory situation be related to the different anesthetic agents used and the effect of these agents on different receptor ion channels?
Hypoxia is a very interesting question. We've looked at hypoxia, and we're continuing to look at it. It's very interesting in two aspects. One, oxygen really isn't the sensor in the body. And I know the Nobel Prize a couple of years ago was in oxygen sensing, or at least I think it was. And I need to look at that more carefully. But for the most part, CO2 is what the body senses when it wants to know about its oxygen levels. And so I think there's some confusion there because we've done CO2 anesthesia and then removed oxygen, and we can get down to 1% oxygen and versus 20%. So getting rid of 90% of the oxygen, and it doesn't knock out flies. So I don't think hypoxia is necessarily an anesthetic. And I think that a lot of the interactions with changes in oxygen are probably the reaction of the body trying to get more oxygen because of how CO2 is behaving. And we do think that CO2 is the first gaseous anesthetic ever used. So my guess would be it would be the fact that CO2 is being manipulated, and that's what's interacting with the anesthetics. But we're also working on that. We'll have more data on that later too.
There are a lot more questions, but since we're running out of time, I will probably pick one more question, and I apologize from others. They can probably email you their questions, I guess.
Feel free to email me directly.
Yeah. So the next question is by Jansu, saying that I was wondering what does make the huge effect in difference of isofluorescent and PLD2 inhibitor?
Isoflurane and PLD.
Yeah.
In the PLD assay?
I'm not sure, I guess.
So, I mean, the PLD2, I must not be understanding. I mean, the PLD2 inhibitor inhibits PLD synthesis or inhibits PA synthesis. So there is no product. So you have no signal. Isoflurane is activating PLD.
Yeah, maybe it's a misformulation of the question, but if Jansu wonders,
I understand it. Sometimes it's just. Can you read it one more time?
Yeah. I was wondering what does make the huge effect difference of isoflurane and PLD2 inhibitor?
The amount of product being the activity of PLD would be my guess. Yeah.
Okay, there are a lot of more questions. Please email Scott, but we are running out of time for now. Thanks a lot, Scott. That was a pleasure to have you.
It's a fantastic series. I really appreciate the opportunity.
Thank you very much. So let's keep in touch. Thanks a lot.
All right, you too. Thanks.